Onto item 7, Nutrients and Red Tide State of the Science Symposium. Will be presented by Betty Staugler of NOAA, the Harmful Algal Bloom Liaison from Florida Sea Grant. And the purpose of this is to summarise outcomes, key insights and recommended research directions from a State of the Science Symposium focused on examining the current scientific understanding related to nutrients and red tide in Florida. Connection to the CCMP. This relates to Water Quality Improvement Action 5.1, support harmful algal bloom, including macroalgae research and monitoring, and measures to proactively reduce their drivers through reduction of anthropogenic nutrient pollution and other contributors. With that, over to you, Betty.
Do you want me to present from here or somewhere else?
Wherever you're comfortable doing it. And I can even do the clicker.
Perfect. Wonderful. Okay.
Sounds good.
So I'm not speaking to the back of your heads. All right. Yeah. So thanks for the invitation. I am Betty Staugler. And so, yeah, last year, my colleague Lisa Krimsky from Florida Sea Grant and I organized a 2.5-day Red Tides and Nutrients State of the Science Symposium. The symposium was at the request of the Florida Harmful Algal Bloom Task Force, and it was funded by the FWC FWRI HAB grants program. And our scientific goal was really given to us. This was our directive from the HAB task force. They wanted to assess the current state of knowledge. They wanted an opportunity for researchers to share current recent findings, ongoing research, and they wanted to identify research needs that could help them in their red tide management as they set priorities for funding opportunities moving forward. Red tide is an interdisciplinary problem, so we took an interdisciplinary approach with the symposium, particularly since we were talking about red tide and nutrients. I will point out this is the first time, to my knowledge, that we had a forum of this kind to discuss these types of issues. We did have diverse participation. About 50 people participated. It was invitation only. And that was because we wanted to make sure we had the right mix of folks in the room. So we didn't want any one voice dominating. The symposium framework, we had 3 sessions: nutrient dynamics across bloom stages, tracking nutrients through systems, and management and response. Our framework was, we started with 1 to 2 synthesis talks. These were longer. And then after that, we would have shorter case studies. These were lightning talks, about 5 minutes. And then what followed was these plenary discussions. These were facilitated, and they focused on 3 questions. What do we know about this topic? What do we need to know, and why don't we know it? From that what do we need to know went into breakout sessions, and we further refined those into questions or statements, I should say statements, which became our research needs statements. Our symposium output is a white paper, and I will have the QR code and link on the last slide for this. And it summarizes basically everything that happened in the symposium. So I do want to point out this was not a symposium where, you know, we had presentations, Q&A, and then everyone went home. We expected everyone in the room to participate and participate actively. What I'm going to share from here is some of the key findings. And I do want to point out, I was the co-chair of the symposium. I was not an expert in the room. So I'm summarizing what other people said, and you know, I used this to co-author the white paper. But at the end of the day, when you have questions, if it gets into the nuances of what they presented, I'm not going to be the one that answers the questions. I would prefer somebody write them down. I know who the presenters are, and I would go back to those presenters. Okay. So to start us out in our first session, Cindy Heil and Kate Hubbard, Cindy with Mote Marine Laboratory, Kate Hubbard with FWC, really kicked us off by looking backwards, recognizing that red tide research has been going on for decades. And we really need to recognize those early efforts because they are what helped set us up for what we know today. And so what kind of the key take home there was that early research focused on the classical nutrient phytoplankton models where inorganic nutrients drive phytoplankton blooms. But those early models fell apart with Karenia brevis, which is a very slow growing dinoflagellate that grows or initiates in oligotrophic waters. And so this became known as the oligotrophic West Florida shelf paradox. And it led to a kind of a new thinking, if you will, from what nutrients are available in the water column to what nutrient forms can Karenia brevis assimilate and use across its life cycle. So look to bloom initiation. We increasingly understand that bloom initiation is more of a coupling of physical and biogeochemical processes rather than just elevated nutrient levels of some source. We know that Karenia brevis cells exist year-round in background concentrations. They take advantage of diel vertical migration to take advantage of the sun during the daytime and nutrients below the pycnocline at night. We know some moderate upwelling is required for bloom initiation. However, too much upwelling favors other taxa that are faster growing, like diet. We know that some organic nutrients are important during bloom initiation and early growth. And we see this with hotspots of dissolved organic nitrogen and phosphorus at the 50-meter isobath. Submarine groundwater discharge provides reduced forms of nitrogen, dissolved organic nitrogen, dissolved organic phosphorus, and trace metals. And then periodic inputs of estuarine and riverine flow it introduces additional nutrients. Atmospheric deposition, such as iron-rich Saharan dust, creates another source of nutrients when it coincides with trichodesmium blooms, which creates new inputs of nitrogen upon senescence. And advanced tools such as isotopic tracing and flow cytometry are starting to really help parse out some of those different forms and sources. When we look at growth and maintenance, we know that Karenia brevis can utilize many different forms of nitrogen and phosphorus, and it can use these different forms simultaneously across both from the offshore to the inshore and across its entire life. The mixotrophy, such as the ingestion of Senecacoccus, provides an additional source of nutrients, particularly during inorganic nutrient depletion. And so it becomes very important during those winter months in particular. And the microbial loop regenerates ammonium, and it provides essential vitamins such as B1, 7, and 12. The physical processes such as fronts help accumulate cells and can create densities beyond what growth alone would be able to predict. And climate variability is really playing a role, we believe, in terms of altering temperature anomalies and nutrient delivery into the system, which influences maintenance. So from here, I want to share a few of the slides and case studies that were shared during the symposium. This slide was presented by Cindy Heil, and it really talks about what I like is she says, you know, these are the nutrients Karenia brevis needs. And under it, these are nutrients that Karenia brevis can use. And we see the suite of inorganic, organic, and particulate nutrients. Below it, she provides some studies as evidence for nitrogen uptake, as well as mixotrophy of direct ingestion of Senecacoccus by Karenia brevis. And here, this slide was put together by Angie Knapp, who is now with Oregon State University. And she very simply says, HABs want to be where the nutrients are. It makes perfect sense. And where are those nutrients? Well, measurable inorganic nutrients and trace metals are right along the West Florida shelf. And dissolved organic nitrogen and dissolved organic phosphorus and trace metals tend to be in the nearshore environment. And then what are the sources of those on the right? Upwelling, bringing the nutrients onto the West Florida shelf, and submarine groundwater and riverine and estuarine inputs, bringing them out into the nearshore environment. So since this is the CHEDP, I thought I would share a couple of case studies and particularly focus on this. This slide was presented by Pat Glibert from UMCES, and at the time she presented, the data was in review. It has since been published in the journal Harmful Algae, so I felt okay to share it. Here she is saying that increased temperatures and increased flows are leading to more severe and longer blooms, and that knowledge can help us predict bloom severity and duration. And so if we look at the graph on the left, we're looking at on the y-axis annual bloom severity. This is based on Stumpf et al. 2022, where the authors created a matrix, a 1 to 6 scale, based on different bloom severity metrics, such as cell counts, toxicity, geographic range or extent, and bloom duration. And on the x-axis, we have temperature anomaly on the West Florida shelf. She broke it out over 2 time periods. And what we see is in this earlier 1970 to 1994 time period, there's no relationship. But as we move from 1995 to 2000, there is a direct effect. And then on the right, we are looking at annual bloom duration and Caloosahatchee River flow. And again, we see no relationship in the earlier time period and a direct relationship in the later time period. And I'll point out the squares are the last 5 years in this time period, so 2050 to 2020. She also looked at nitrogen loads associated with the Caloosahatchee River flows, and in the paper broke it down by months and determined that the direct effect comes with a 3 to 6 month lag. So the authors proposed a new model for Karenia brevis growth. And so here we see during the late summer and fall, Karenia brevis has great water temperature. It's able to take advantage of those flows and nutrients associated with them. We also have a lot of summer activity out on the shelf that's bringing nutrient waters to the West Florida shelf and some microbial activity. During the winter and early spring, temperatures are cooler, but we're starting to see those warmer temperatures. Karenia brevis typically takes it, shifts to internal recycling of nutrients during this time period, or external nutrient sources as a result of mixing from coastal fronts. This is when the blooms typically peter out, but because of that delay and in the warmer temperatures, we are starting to see more blooms survive this period, and if they do, then they have ample nutrients available once the rains start again. So here I'm going to talk about submarine groundwater a little bit. This was a presentation by Chris Smith from USGS. And the slide that was right before this, can't think of the pre-word that I'm looking for, said, think everything stops at the coast. And then he flipped the slide and it was not exactly. Seabed and aquifers are offshore sources. So on the we're looking at benthic exchange and the three primary forms of benthic exchange: waves, which force water into the sediments as a result of these concentric circles that they make. And number B is currents across the seabed, which induce flux across the seabed as a result of these high and low spots. And then C is waves, which compresses the aquifer during the high tide and then releases water into the water column during the low tide. On the right, he's showing the water cycle for the purpose of showing that groundwater discharge can impact or can be released in the inshore, nearshore, and offshore regions. So Chris presented this slide, but I from this Kurtz et al. Paper 2023, I pulled this picture out of the paper for this presentation because it does focus on the Charlotte Harbor region. So these authors did groundwater tracing studies from Anna Maria to Ave Maria. They were particularly interested in Lee County because there are more individual aquifers under Lee County than anywhere else in the state. And the aquifers that apply to this, this image are the superficial aquifer system and the lower Tamiami aquifer, which are both recharged by precipitation. They had groundwater wells and they did tracing studies, and they determined that the superficial aquifer is a source in the tens of meters of inshore submarine groundwater discharge. And the lower Tamiami is a source in the thousands of meters of offshore submarine groundwater. And so this hatched area is roughly what the lower Tamiami, that area would be. On the right is they mapped bloom severity against precipitation for each county. So this is Sarasota County. There's also one for Charlotte, Lee, and Collier County. Bloom severity here is simply the average cell counts divided by bloom duration. The blue dots are when we did not have severe years. So this is just your run-of-the-mill bloom, and we see very little, if any, correlation between precipitation. And then the orange dots are when we had severe blooms, and you see a direct correlation between precipitation and bloom severity. The authors hypothesize that this is because during most years, nutrients associated with submarine groundwater discharge are tied up in the nutrients or in the sediments as a result of dissimilatory nitrate reduction to ammonium or DNRA. However, during some years when we have higher precipitation, DNRA fails and releases more nutrients into the water column, making them available for Perennia brevis maintenance. Uh-oh, this is not working.
I'll advance it for you.
Okay.
It's not working on my end either. Okay, there you go.
And I will say, you know, they hypotheses, so more work is noted. All right, so in session 2, we talked a lot about episodic events and feedback loops. And this was a great slide that was presented by Ed Sherwood from the Tampa Bay Estuary Program, where he looked at the Piney Point discharge and its relationship to perennial brevis. So here on the top, he's looking at that initial nitrogen response from the discharges. And we see the immediate response was from fast-growing diatoms. And then that was then replaced by pretty fast-growing benthic cyanobacteria blooms. And even though perennia brevis was in the lower bay on April 18th, it really didn't get a chance to get a foothold until those blooms had dissipated. So it just kind of shows there's a kind of a right nutrient amount, and there is always competition within the system. Oh, wait a minute, I went too far. Okay, this was presented by Young-Gang Blow from USF, and he was talking about loop current forcings and how when we have strong loop current forcings, it will create stratified conditions where we can measure that with temperature. Because we're seeing the deeper, colder waters, and it's bringing deep water nutrients onto the shelf. And this often occurs when we have our tropical systems. And so this image on the right is showing what it looked like after Hurricane Ian, where we had nutrients affected onto the shelf that collided with all of the rainwater and nutrients that were coming out of the estuary. This is some images from Chen and Lee, 2024. The image here shows basically the same thing. They do point out, and I didn't point out earlier, wind from the north is what favors upwelling. Wind from the south favors downwelling. So upwelling is going to bring your cells and your nutrients onto the shelf. And on the back side of the storm, because they're counterclockwise, you're going to get that north direction wind. So this is what it would have looked like in a model after the hurricane. I'm just going to focus on these down here. This is looking at the immediate response. So here are our nitrate, ammonium, and phosphorus is the black line. And you can see the immediate uptake was by diatoms and Senecacoccus, and then later by perennium brevis. So eventually we have to get to decline, right? The blooms have to end. And this is arguably the part of perennia brevis research that is most lacking. And a lot of that is because scientists just can't be there for bloom collapse. But we do know that it's rarely just a single mechanism. It's often a combination of things that lead to bloom decline. And that can be nutrient exhaustion, which stresses phytoplankton, physical forcing, which moves cells out of the area away from their nutrient source or alters nutrient ratios. I'm not going to get a chance to talk at all about the biological antagonists, but there were some great slides that talked about the role of viruses and algacidal bacteria. And some of you came to the DynaShield workshop and know that we're looking at algacidal bacteria for bloom control. And then importantly, after a bloom, succession usually starts with diatoms again. Although some Perennia brevis pockets will remain, and we see those in background conditions, which is what helps we see those new blooms when conditions are right. So, after a bloom, we typically all go back to our, whew, we survived another one. But Chris Anastasio reminded us that sometimes blooms can have long-lasting effects. And so this was from his synthesis talk during our second session. And here he's showing from tracer studies, the counterclockwise circulation patterns in Charlotte Harbor. And so we would expect that biomass from fish kills and marine mammal mortality would move up and be along the eastern shoreline of Charlotte Harbor. We know from reporting that approximately 2,000 tons of fish was removed by Charlotte and Lee counties during the 2018 to 2020 bloom. And Chris did a little back of the envelope calculation, assuming that for every fish that was removed, another one remained in the water, and came up with an estimate of approximately 13 kilograms of dry weight of nitrogen that would be available in the system after the bloom. And I think most of us know that there was a pretty immediate response after the bloom with Clerpa fastigiata. And between 2018 and 2020, significant seagrass loss harbor-wide, but mostly on the east wall where approximately 50% of the seagrass was lost. And so Chris did another back of the envelope estimation of what the seagrass decomposition would contribute in nitrogen load to the system as well. So I mentioned that after our presentations, we went through this process of what do we know? What do we need to know? Why don't we know it? And we did this for the first two sessions, the nutrient dynamics across bloom stages and tracking nutrients through sessions or through systems. I'm only showing snippets of what we don't know for the different bloom stages. And a lot of it is we really don't know the actual roles of these different sources. And so that's where a lot of the isotopic tracing work is needed. We have a difficult time quantifying recycling. How much is it? How, is this nitrogen or is this, from 100 years ago nitrogen? And then biomass state with 2 Bs apparently. And just not being able to be there for those bloom events. The folks in the room also recognized that there's a lot of work that's being done, but we need to collate some of that and have more comprehensive field studies. So once we went through this process of identifying what those gaps were, these are written up in the white paper in paragraph 4. We also put people in rooms. So we had 10 to 12 people each in a room, and their job was to take those research needs and develop research needs statements. And so you're seeing just a very small snippet. There's actually a whole bunch, like 3 pages worth of research needs. Now, our last session was management and response. And here we had some great talks that talked about legislative updates. DEP talked about their water quality monitoring programs related to TMDLs. We had a talk from FDACS who talked about FDMPs, and Charlotte County talked about their septic to sewer initiatives through their One Water, One Charlotte program. We also had talks from FGCU and SCCF who talked about how their research is supporting state and local government needs. And instead of doing the what do we know, what do we need to know, why don't we know it, we did a different activity. And this is because the HAB Task Force's mission is not to tell Charlotte County how to do management or DEP how to manage water quality. And so we really wanted to do an activity that would help align some of the research needs from management back to those first 2 sessions. So we asked people to envision what a healthy water body would look like if we could achieve that in like 25 years, and what characteristics that would achieve, and then what needed to happen to achieve those results. And that allowed Lisa and I to go back in and make some of those connections. And so I want to share this slide. This again is research needs. And I don't know, can you guys see the individual research needs? I was hoping it was big enough. I picked this particular section because I think this is relevant to this conference. And I wanted to point out, because we often think research needs as being, like core research, but these are really research and management needs, if you will. And so I do encourage you, when I give you the link, which is on the next slide, to take a look at that. And because I think it's important, and I think it speaks true to the CCMP and what this conference tries to achieve. I mean, even defining what a healthy water body is, which we could not agree on. So that's important. And with that, I'm going to share the QR code. I made it nice and big. I do encourage you to check it out. The white paper it's really divided into 3 primary sections, which are basically the sessions, the 3 sessions. So you're going to read what we know, followed by those gaps in understanding. At the end is the research needs, and they're just kind of listed out like I showed you on the table. We also did some additional activities where we tried to identify some of the monitoring activities. So those maps are in there, and how you can access maps to see who is doing what types of monitoring. But the primary guts of the white paper, I would say, are those 3 sessions. So thank you.
Okay.
Thank you.
Thank you, Betty. Any questions or comments for Betty?
Well, people are formulating their thoughts on this and questions. I wanted to mention that the last cycle at our management and policy meetings, we had a harmful algae bloom theme, and we had Lenny Lindau come to talk about red tide in terms of looking at data from the Lake Okeechobee Caloosahatchee system and nutrient loading. There's been various papers looking at, as you know, that one that Snepp helped to co-author was looking at nutrient loading from the different river systems related to severe long-term red tide events. That paper and fact sheet is on our website, but he took it a step further and continuing to kind of do various analyses of that and presented that at our last management and policy meeting. So those presentations are available, those videos, on our committee web pages, as well as on our YouTube. I had to mention that in case anyone was interested in this topic, to who might want to go check that out as well. I thought, and is particularly interested in that system.
I do want to point out, Lisa and I took great lengths to write the white paper in a way that wasn't so overwhelming that nobody would want to read the white paper. So hopefully you guys, if you do read it, you will enjoy it and not go, oh.
Yeah.
Thank you.
You're welcome.
We're looking forward to that.
Yeah.
So I know that there's the Florida HAB task force. I believe there's a federal task force, and I've listened to meetings of both, and it sounds like both are doing the same thing, but is everybody communicating together across those task force?
Yeah, so I think what you're referring to is there's the HAB task force, which is kind of led by FWC, looking at marine HABs. And then there's the blue-green algae task force, which just met yesterday or the day before, which is strictly looking at freshwater
But there's also a federal task.
There is the,
And it's
sort
not,
of,
yeah, but
sort
it's,
of.
yeah,
There
it's
is,
a longer
yeah,
name.
so there's
to it.
HAB, and I participate on the interagency working group for HAB, which has not been,
Yeah, that's what I-
the last year. Due to other reasons. But yes, so yeah, the interagency working group, or actually the HAB, the directive is to NOAA to work with these different federal agencies. And so part of that was creating this interagency working group. And David Kidwell from NOAA is the chair of the interagency working group, and he regularly participates in those task force meetings in Florida, as well as just directly with FWC and FWRI to make sure that there is some communication. And I think you probably participated, like when we were doing South Florida assessment, which unfortunately kind of dwindled when the change of administration happened. But yes, I think there is quite a bit of effort to communicate. And in fact, we invited David, who was not available, but Bridget Weimer from NCAUSE did give a great presentation at the State of the Science Symposium on what was happening at the federal level. That's a good question. Thank you.
Locally, as sort of a subgroup to the TAC, we have the Charlotte Harbour Algae Working Group, which obviously Kate is sort of a driver of, and I think it'd be good for that group to pick up some of these ideas and see what applications there are at that local level here.
Yeah, I'll second that sort of. It's interesting seeing these developing or hypothesized links between the red tide and other coastal eutrophication problems, with the dead fish causing macroalgal blooms, maybe, and other coastal nutrient sources maybe then contributing again to the red tide. And the hangover effect in recycling of nutrients is something I think we've hypothesized for a long time, but to see some of that science taken further should be interesting.
Yeah, I mean, I think for me, at the symposium, the submarine groundwater discharge, I really thought that was an interesting concept. We don't talk or hear about it enough. Even, maybe some of we know, sometimes it's mentioned, but we don't really see the science behind it. And so I thought that was really interesting, which is why I wanted to share a couple of those studies with you guys. But one of those studies, there are probably far more questions because again, we're hypothesizing and we really need a lot more research to really get to the point where we can pinpoint those sources, and that work is starting thanks to some of those new technologies. So
I know a lot of municipalities are doing deep well injection. And based on one of the slides, it seems like it's all connected. It's going to bubble up there somewhere. That's being examined too, right?
I don't know the answer to that question. I would suspect that somebody from USGS would have ideas on that. It's something I could ping past Chris if there's interest.
Well, normally they're injecting below the aquifers you were thinking about, much deeper. So, I know that even in the boulder zone, eventually it meets up with the ocean. So even things that are injected that deep can find their way laterally out into the open ocean, but they're theoretically lined, in a manner that would not allow them to intersect any of the aquifers used for drinking water.
We don't know.
Yeah.
Yeah, but I mean,
Former-
like I said, I'm feeling pretty good about presenting what was shared at the symposium, but the nuances, I don't work in deep water or submarine groundwater, so.
The predictive succession model there, is that at the stage yet where it can be used in sort of emergency planning, emergency response, where if we start to see increases in the diatoms or whatever, counties and cities can start putting their action plans into effect for red tide response?
Actually, a really good question. I don't think there's anything that's. I mean, there was a lot of discussion about the need for more comprehensive models, needing data and those types of things. Are you referring to? Which was back here.
Yeah.
That.
Maybe even the one couple slides where you had the Ed's paper that had the succession. Yeah, here. So we have that kind of the diatoms come first, then the cyano. Wonder if there is a way, if we start to see these patterns playing out, we know that maybe 6 months down the track.
Yeah, I mean, it seemed like that held true. In the instance where diatoms came first and then perennigraphists. In the offshore environment, you're not going to have the same benthic cyanobacteria bloom, but here, where you have the immediate response being diatoms and Senecacoccus, and then later. So I mean, I think we heard that over and over again, but whether there is an actual model that is helping to predict that, I don't know.
So I know, of course, it's very important to continue to research and tease out these connections and make all of the cases as to what the different contributing factors are and whatnot. But are you also then bridging that into like management and policy recommendations? I know one of the things, like a couple of years ago, when we did have a red tide event, we brought in like Pinellas County, who has like a standing contract with a contractor to, like, as an emergency response after a red tide event, go out and immediately try to physically pull the fish out of the water or off the beaches as quickly as possible so that it doesn't cycle. And we were sharing that with other cities and counties at the time, saying this is something practical that people could put, into effect so that they're more prepared the next time, to react and be able to do something to try to lessen the impact or lessen the severity and duration of the event by just doing what they can,
Right.
to take away a contributing factor. So is that part of like what you all will be doing, is like codifying any ideas like that into a document to like give back to cities and counties as to what they might be able to do?
We have not been asked to do that, and CGRANT would not do that independently. I know FWC during blooms works very closely with, they have like a mechanism which was based on a, CGRANT project, the Red Tide Communication Plan, where they communicate with local governments, and then they communicate with, visitor and convention bureaus. And so they have a mechanism in place, and I would think that would be more likely to come through that process.
It just seems like a toolbox of some sort would be really helpful,
I do know the Red Tide HAB Task Force Communication Working Group has been working on a lot of different outreach types of materials. I don't generally attend those meetings, so I don't know what they are working on, but I would think that would also be a place where something like that might come out of. So, so that's my way of saying I'm really not sure.
Okay.
Any questions online?
Thank you.
No, thank you very much.